Alrighty, let me know if you guys can't see my screen at all at some point or something messes up. But hi, I'm Nahal Khalkhali. I'm a fourth-year neuroscience major. My research is about the study of the role of the Cas family adapter proteins in the migration of precerebellar neurons. I worked with Dr. Martin Rico Mango. Um, And so some background information before we begin about the Cas protein and the Cas gene. So the Cas family of signal adapter proteins, if you can see the picture right here, we have an integrin receptor. And so basically the integrin receptor activates, causing Cas right here to become recruited by both FAX, paxillin, and SERC. Cas is then hyperphosphorylated and remodeled into a Cas-Kirk-Doc 180 complex that goes on to cause actin remodeling. And this makes Cas important for the cell adhesion, proliferation, and migration events that occur. So some background information about the neuronal migration pathway that we'll be looking at. So the neuronal migration path that we're going to be looking at is going to be the AES right here. So neuronal migration is based upon proper guidance of molecules, and they can get cues from both short range and long range cues. So the migration can either be radial or tangential. Tangential is going to be glia independent, and it's going to be parallel to the surface, while radial is going to depend on glia scaffolding, and it's going to be perpendicular to the surface. So as I said, we're going to be focusing on the AES stream right here, the anterior extramural stream during precerebellar development. The AES stream is going to be a tangential migration stream that helps relay messages of coordination and control of the automatic systems of the body. So some methods and materials that we used to figure out the impact of the Cas gene. So we specifically use the CRE-LOX system. So the CRE-LOX system, we use this to remove the Cas gene and see the impact that it had on pontine neuron migration. So we use the CRE gene right here to come, and it specifically cuts at the LOX P sites, which are represented by these blue triangles. And by cutting at these LOX P sites, it allows for the deletion of specific genes. And in this case, we specifically wanted to delete the Cas gene. And so we developed different CRE driver lines to see the Cas family function and what would happen when we deleted the Cas gene. So these are some of the CRE driver lines that we developed. There's the nestin CRE, and we use that to broadly assess the role of the Cas genes. And so the nestin CRE deletes the Cas gene from both the, as you can see, the neurons in blue and the glia cells in green. While the Math1 CRE we use to assess the neuronal autonomous role of the Cas gene, because the Cas gene was deleted specifically only from the neurons. And then in future work, we're going to be using the human GFAP CRE-ERT to assess the glial autonomous roles for the Cas genes compared to the Math1, because this CRE driver line deletes the Cas protein from only the glia cells. So the results that we got from doing this. So we determined, So we determined the expression of the Cas gene during hindbrain development first to see if Cas was present in hindbrain development. We used the GFAP to determine the expression of the Cas gene, and we saw that you could see kind of in the green and how it migrates upwards, that we saw that pontine neurons express this Cas gene as they travel through the AES stream. So now that we know that the Cas genes are present in the pontine neuron migration path, we wanted to see what would happen if we were to remove the Cas genes with the CRE-LOX system that I was explaining before. So first we removed it from both the glia and the neurons using the nestin CRE. And we saw, to see the requirement, we saw that in the E14.5 right here. The nestin CRE, where it's both the Cas gene is removed from them both, we saw that there was an error in the pontine neuron formation, as you can see with the red arrowhead right here. This is the glutamate control, and then this was the mutation. And then in the E16.5, we could see some dramatic stalling of the pontine neurons in the midline. However, there was no disruption in the other nuclei that are formed during the hindbrain development. So now that we know that there was an impact when CAS was removed, we wanted to specifically see where this impact was. Was it glia or was it neuronal? So we removed the CAS gene from the neurons only to see if there was a neuronal autonomous requirement of CAS. And so we saw in the E16.5 that, and also the P0, that the pontine neuron formation was pretty similar to the littermate control. There was no dramatic difference, and it seemed pretty similar with no error. So therefore, we figured out that there was a lack of neuronal autonomous phenotype. So we decided to investigate the function in the radial glia and the surrounding glia. So now to test that function, we first had to see if the CAS function, if the CAS gene was present in the surrounding radial glia. So what we did was we investigated the expression in the two different radial glia populations. So the first population we checked was this population, the BLBP radial glia. And we were able to see that the pontine neurons expressed CAS as they traveled laterally to the pontine nucleus. And the second population that we tested for the radial glia is the RC2. And the RC2 population illustrated that CAS was present in the end-foot processings of this radial glia. And overall, it just showed that that CAS is present in a diverse population of radial glia through appropriate temporal windows, and they play a role in the pontine neuron migration. So some future work is we're going to be focusing on the radial glia population. So since we figured out that CAS is present in this radial glia population, we want to use the CRE line driver that I explained before to remove the CAS gene and see the impact and test the non-autonomous roles of the CAS proteins with the new CRE driver line, and to remove the CAS function from that radial glia and analyze the phenotype and see if it had an impact on the pontine neuron migration. So some questions that we have about this is, which subset cell population is required for this gene for pontine neuron migration? Currently, we believe it's the glia cell subset population. And then if we test this out and we see that there is no impact on the migration with the radial glia, then CAS definitely has an impact. So maybe there's another molecule that's playing a role alongside together with CAS in the signaling path cascade. And then some questions I had was, how is this mutation impacting the animal behavior? So currently all the animals that we have that have this mutation, they seem to be behaving normal. We haven't run any tests about them, but that would be an interesting thing to try out in the future, and to see how this mutation is impacting the neuronal connectivity with other areas of the brain. Since the pontine neuron is so important and it connects with a lot of different areas of the brain and plays an important role in motor behavior and balance, maybe us mutating that area for the CAS genes has an impact with its connections in other parts of the brain, maybe weakening or strengthening that connection. Thank you so much for listening to my talk. I'd like to thank my lab members and my PI, Dr. Rico Mango.
Okay.